So we've been talking about double-blind placebo-controlled trials and how simple and elegant they are, but also how this simplicity doesn't mean that they are always flawless. And I've been looking at some of the issues involved in them over a few posts now. Today I'd like to talk about significance levels. There, I can probably stop now because nobody is still watching. Let's all go off, have lunch. Oh, okay. Well, I'm glad to see you're still here. So, listen, in this kind of study, like many studies, we've got two conditions. In this case, one group gets the real drug we're testing. The other gets a placebo that looks exactly the same, but has no active ingredient. And we're looking to see if there's a difference between the two. So we measure everyone at the beginning, measure them again at the end, and subtract one from the other to get a difference score. And we compare the different scores for the drug condition to those from the placebo condition. What we're hoping is that the drug condition produces a bigger effect, a bigger difference from before to after than the placebo condition does. So if our study is on an antidepressant, it's possible that people taking the placebo will get somewhat better, either because of expectancy or because of the simple passage of time. But we're hoping that the people taking the antidepressant get a bigger benefit than that. But how will we know? Let's say the measure we're most interested in is improvement on the Hamilton Rating Scale for depression. The placebo condition, let's imagine, produces an average reduction of depression of 3.427 points. Now, what are the odds that the other condition will produce exactly the same change score, even if the drug does nothing and is really just another placebo? Well, answer, approximately 0. It's not going to happen. By sheer chance alone, these groups are going to be at least a little different. So if the antidepressant condition gets a change score of 3.500, that's no real surprise. We want to know that the drug is so much better than the placebo that it's unlikely that difference is due to chance alone. So we do a statistical comparison, usually at the 5% significance level. That means that if we assume these two pills are no different, we'll get a difference between them that we shouldn't get by chance more than 5% of the time, or 1 time in 20. If we get a bigger difference than that cutoff, we can say that it's pretty unlikely the drug is a complete dud. We probably got that difference because the drug is actually doing something. So if the difference between the two conditions is bigger than that, we say that it has achieved statistical significance. And in most clinical studies, that's what you're looking for. And depending on how variable the data are, it's possible that if we get just 1 point better for the drug than the placebo, we'll reach statistical significance. So if the placebo gives a 3-point reduction in the depression score, and the drug gives us a 4-point reduction, we may reach statistical significance. And in a lot of drug studies, we do get that kind of improvement. It gets past that 5% level. Barely, but it does. Well, so great. If the controls in the study are working properly, we may be discovering a genuine effect of the drug. And after all, that's what we've been looking for. But there's a problem. If we get 1 extra point of decline in those depression scores, nobody cares. Go up to everybody in the placebo condition and ask, "Are you better?" They'll say, "A bit." Go up to everybody in the drug condition and ask, "Are you better?" And they will say, "A bit." Same bit. Even they can't tell the difference. It's a difference big enough to reach statistical significance, but it's not big enough to make any real difference in their lives. Take Alzheimer's. We'll often assess Alzheimer's with a mini mental status exam. It's a 30-point test administered by a clinician asking the person about things like the day and date and a bunch of other things. It's pretty easy if you have a functioning memory. So most of us would get almost a perfect score. As Alzheimer's progresses, people generally get lower and lower scores. And let's say you come up with an anti-Alzheimer's drug. We don't expect the score to get any better, but maybe it won't drop so fast. Maybe people's function won't go down as quickly if the drug is effective. So let's say both groups, placebo and drug, start out with a score of 21 out of 30. After 6 months on the drug, the placebo group has dropped to 15, 6 points down. The medication group has dropped to just 16.5, 4.5 points down. Now that's a difference of 1.5 points, and maybe that's enough to reach statistical significance. But if you ask the ward staff, who do you think got the real drug, Group A or Group B? Chances are they won't know. It's a difference, but it's not big enough to be noticeable. Well, who cares? If that's the best you got, do it until you find something better. Except if you begin factoring in the cost of the drugs and whether the effect actually lasts. And maybe the side effects, if there are some, maybe the downside is bigger than the upside. Many medication studies look for statistical significance, but that's not really what we're after. We want clinical significance. That's noticeable improvement. We want better quality of life, something big enough that the person really notices a difference. This is the kind of improvement that we get with vaccines. We might start with practically everybody vulnerable to measles if they get exposed. And then we vaccinate them, 97% of them are immune. That's a huge effect. We dream of something that effective in mental health. When we start looking for actual clinical significance, our treatments wind up looking much less impressive. We do better than placebo, but not by much. How could we correct this? Well, to some extent, we're already doing it. In a lot of medication trials, they look at the number of people achieving a bigger change, something we define as a clinically significant improvement. But it's not uniformly done. And the bigger the change you're looking for, the less likely you are to get it. So even these clinically significant improvements aren't always that ambitious. So this is another potential challenge for the double-blind placebo-controlled trial. What do we count as a success? Is it statistical significance? Or is it a genuine, marked change in the symptoms to the point that the person is really functioning much better? Part of the problem is that what counts as clinically significant isn't easily defined by science. It's more of a judgment call. But the more we focus on this question and treat statistical significance as only the first step in demonstrating treatment efficacy, the better we'll be able to guide clinical decision-making. If you like some of these ideas, my book, How to Be Miserable, 40 Strategies You Already Use, is available from any bookseller, online or in person.